Hello, little fishies. No, I am not in an aquarium. I am in one of the labs at NCBS. Hi, I am Sonal, the Communications Officer of NCBS. And on the special occasion of National Science Days, we are going live from one of our labs. So right now we are in Dr. Vatsala's lab, and it's all about neuroscience here, and it has a lot to do about fishes. So let's see what it is about, and let's meet one of her students, Shivangi. So who's very busy with a microscope. Hi, Shivangi.
Hello, hi.
Yeah, tell us about yourself. What are you working on?
I am a PhD student in the lab.
Okay.
So we call ourselves the Neural Circuits and Developmental Lab, headed by Dr. Vatsala Thirumalai.
Okay.
And the broad aim of our lab is to understand how brain generates movements.
Okay.
So,
How brain
generates and controls movements.
like any movement, like walking.
Like walking, dancing, you are talking, your muscles are moving. All of those muscle movements are controlled by brain, and we are interested in understanding how does that happen.
Great.
So, like you saw, fish over here. These are adult zebrafish, which we use as our model organism.
Okay.
And we basically look at the young fish, which are babies of these adult fish that you see here.
Okay.
So if you can come here, I'll show you.
Sure. So this is in some special microscope?
It is a microscope that is just helping us visualize fish at some magnification.
Okay.
So if we look here, this is a fish that is freely swimming, and if I give it some stimulus, it will execute some movement. Right? You see that?
Wow.
So movement is what
here,
we want
right?
to study.
So tiny, like I can't see it nicely with my naked eye.
It's a few
Wow.
millimeters in size.
Oh, really?
So, yeah. So like you're seeing over here that the fish is moving. We are also interested in now how is the brain generating these movements. What neurons in the brain participate in, executing such
The neurons, the cells that are present in our brain,
Exactly.
right?
Neurons are the cells that are present in the brain. So to be able to do two things simultaneously, where we also see the fish behaving, doing some form of movements, and also be able to record what's happening from the brain, we have a different kind of preparation that I'll show you here. So if you take it here, so now here we have restrained the brain or the head of the fish.
Okay, this line that we are seeing. Right?
Yes. So
This
that line is the point beyond which everything beyond that point is restrained, and everything
so
below
that won't move.
yes.
Okay.
And everything below that will move. So that's the tail, and if I give it any stimulus, it will be able to execute these swims, right?
Okay.
So now we have a preparation where we can see the fish swimming, as well as record or image the events that are happening in the brain. So I can take you to the next room.
So
where
what do these graphs and all mean? They are like some.
So these are just for imaging. I mean, for right now, to be able to visualize it with the right kind of light, those are the parameters, and those are like set for us right now. Yeah. Okay.
Microscope is no longer a simple microscope,
sure.
right?
Okay. So to record events and activities that are happening in the brain, we use something that is called electrophysiology.
Electrophysiology.
Right.
Okay.
And
Thank you.
Poonam is here, and she's going to.
Hi, Poonam. Peekaboo. Hi, Poonam. So thank you, Shivangi. So hi, Poonam. Aren't you a postdoc at Vatsala's lab, right?
Yes.
Yeah. So what are you working on?
So she must have shown you those immobilized preps, larvae, right?
Yeah.
So why we do that? Because if they are moving, it's hard to focus the camera on the brain and record the activity, right? So if you come here, what I can show you is that.
Wow. This is a chamber.
So this is the place where we actually, get the electrical recordings, right? Which can somewhere say that, how those swims that you saw in the prep look like. So actually, if you
So this is a very fancy microscope.
So this is just the simple microscope, and what is happening here is the place where the prep is there.
what are
larvae.
these things? So these are common to all microscopes. You have
Yes.
the place where you keep the
Yes.
sample. But what are these?
So exactly. So that there you saw that if the fish is just moving the tail, the camera can capture it, right?
Right.
But here what we are asking is we are actually recording the activity from the tail as well. Because if the prep is fixed, then we have to, understand the behavior in some manner, right?
Right.
So to record those activities, neural or electrical activities, what you see, these are the you can call them as like manipulator, or these are the electrode holders, right? One of the electrode is going there, in which there is a solution, and when we go and touch the neuron, we can actually record the activities there.
So basically, these instruments connect with the fish's neurons
Neurons, yes.
and
neurons.
record what's happening inside.
Exactly. Records
Wow.
in a sense that because neurons talk to each other through these electrical signals,
Okay.
and that's why we can use this way of recording, because these are connected to the amplifiers here that you see,
Okay.
and it can amplify the signal and show you here. So I can show you one place where you might be able to see the electrical
This
activity.
white line going.
So you see, what you
Oh.
see are the swims, right?
Okay, so these waves are like the swims. that-
Exactly.
wow.
So when actually the fish swims, there are activities that are happening inside, right? These
Right.
are those activities. So what I'm doing, I'm just shining the light, and in response to light, see what happens? So sometimes they get saturated, but if I increase the activity,
So activity as in the fish's activity.
the fish neurons activities.
Okay, how is that changed? Like by showing some light or
So
something?
see exactly here. Can you see it?
Yeah,
came?
yeah.
So when I showed the light, so imagine right now when people are seeing this video, the information is going from their
Right.
eyes, and then there is some activity, something
Going
going
in the
inside their
brain.
brain and telling them then, oh, this is that and that, right? So the same way when the light shines on the fish eyes, the eyes receptors get activated and tells its other body regions that now you have to swim to run or eat or something. So if the fish is running, it's hard to record, right?
Right.
So what we do is we place the fish at one place and then go and record from its brain or from its other areas, because there are
Cool.
the nerves that will be going all over the place, right?
Okay.
So it's just the light that you shine.
So it's like light and like different stimulus you give to
Different
the
stimulus, yes.
fish, and the brain of the fish, like the nerve cells, are reacting.
Exactly.
And that whatever is happening in the neuron, these little
pipettes, these glass pipettes.
manipulators,
Huh,
is that correct?
The glass holders.
Yeah,
yes,
these
the-
glass holders record it and they bring it
send
to,
it exactly
to the computer,
right,
and you
then
see
it's
it
something
as,
like what you see here.
and then this helps you understand why and what is happening in the
Exactly, because now what
Wow.
you can do is you can actually record also from the these swims things, and then at the same time record from their brain cells as well,
brain. Okay.
right? So when the brain cell gets activated, what happens to the movement?
Right.
Both information you will get.
Okay. So what type of stimulus is given? Is it just light or different? things?
No. So I can show you one thing. There is a screen here. There. So
Okay,
what
this
we can
thing,
do?
this thing, right?
Yeah, this screen is there.
Okay.
We can display things here in the screen. So if
So like you can show images.
exactly. We can show images, or we can
Let me get closer to
Yeah.
this.
I can actually
Wow.
Display
Even
one
the
of
fish
the-
is enjoying a nice view.
yes. So yeah. So now your screen is active, and you can see that as soon as the light came there, here you saw the activity, which is the
This
swimming of the fish. Yes.
Okay.
Yes. And now what we can see is I can show you just the demo, because we have to put the screen down and all.
That's amazing.
Yes.
This is the whole processing is happening in these.
Exactly. So these are these are very tiny signals, and you have to put them there to amplify it. Yes.
Okay. So a lot of physics is also involved, right?
Oh yes. Sure. It's all like integrated, physics,
Yeah.
maths.
So something that goes
Computation.
on today's theme about interdisciplinary research, because you
Yes.
depend a lot on
Yes.
like chemistry and physics and bio, and people usually like outside these places we usually think it's just like some bio going on or some chemistry
Exactly.
going on, but it's like, you know, you all have to learn all of this, you have to work with this, you have to understand how
Exactly.
this.
So this
amazing.
education system where we have separated maths, bio and all, you know, at this stage you will feel that actually there is no kind of
Yeah.
separation. You want to learn how you code, so now you can
So you have to do coding as well.
Exactly. So
Wow,
this
you
is a
have to be
code.
really talented to be a scientist.
Oh no, it's not that hard.
Oh.
So this
Wow.
you can see the code which actually projects these gratings there in the
Like
screen.
these black and white things?
Black and
Oh,
white,
we
which you can
them.
see there. Yes.
Okay.
So now when it goes underneath, the fish is actually seeing it, and you can see it's moving there also.
Okay,
So
and
when
then...
it moves, it's like a virtual arena for the fish.
Oh, nice.
So when it moves, the fish also tries to move.
So
right?
like a VR headset that we
Exactly.
see and we react, but it's just like
You are at the
able
same
to-
place. You are
Yeah,
stationary.
sta-
Just the world is moving around
Oh
you.
wow.
And then what happens is, right, when it moves and the fish gets the signal and all, so we can,
You record it here.
then the activity comes here,
Oh.
right? See, that grating moved and the activity came here.
So you have to like use very specific stimulations because this is like millimeter long fish, right?
Yes.
So
you have
it
to
won't be
use-
like two humans, you'll show fish or like pictures of people, things happening. But with this tiny
Mhm.
fish, you have to have like these abstract things, right?
So whatever, make meaning to the fish.
Okay,
So fish,
yeah.
because mainly they swim, so you want to put them in those kind of input to in response to which
Right.
they feel like they have to swim or something.
Swim or something.
Because if I just
So something that generates movement in them, right?
exactly that can
Okay.
generate movements, right.
That's nice.
Yeah.
Wow, that was really amazing, Poonam.
So yeah,
Thank you
so here because you saw these single activities, right? The nerve
Yeah,
recording
yeah.
or the single neuron recording, it's one cell. But in the brain there are so many cells. It's like, a population activity that results to some movement.
So are human brain cells also similar to that of zebrafish? That's why we've chosen that as
yes.
the
They're close to, very
Close
close
to.
to the properties of these. And, the connections that they make.
Okay.
Yeah. So now what you will go there is something that you can actually see the population activity, the activity
Okay.
of many neurons instead of just one neuron.
Okay, so right now we were studying one neuron, and
Exactly.
now we are looking at multiple neurons,
Exactly.
studying them together.
Exactly. Yes.
Or is it like you study them one by one and then you club the data?
You can do that also, but it's good if you can, get
Simultaneously. Okay.
or activity from many neurons.
Okay, so now we move to another interesting room. Let's see what they have for us.
So this is the imaging room,
Okay.
right, where you image the activity in the whole brain of the fish.
Okay, so is this the brain?
Yes.
Wow.
And Harsha is here,
Hello.
Hi, Harsha.
who is engaged
Hi,
into
Sonal.
this, and he will take you through this.
Thank you so much, Poonam.
Thank you.
Happy Science
Bye.
Day.
Bye.
So, Harsha, first tell us about yourself. You are working as a...
I am a PhD student here,
Okay. Yeah.
and what I do is I look at neurons in the brain, all of them. That's not a great picture. I'll show you that
Okay.
once we start the experiment.
Sure.
So they have probably told you about how they record from single neurons.
Yeah, right.
So what we want to do, what more we want to do, is that getting information from one or two neurons is not sufficient.
Right.
So we will look at all the neurons in the brain at the same
Right.
time and see what everything is doing and how the full brain is leading to the
Okay.
behavior that the animal might be doing.
Okay.
So we use this microscope for
So
that.
this is like a very interesting and uncommon microscope that I've seen. Not like those typical like you have two lenses and all. So can you please tell us about it?
So this is a light sheet microscope that
Light sheet
light
microscope.
sheet microscope, yeah.
Okay.
That Sriram in the lab and I built,
Okay.
and this is what we use for our.
Can you tell us a little about it?
Sure.
Guide us through it.
So we have a laser there. That box is the laser,
Okay.
and the light then passes through here, this way.
Okay, so
This is
there's
the
like a
exact-
mirror and then it
Exactly.
goes
Yeah.
there.
So there are two mirrors. It gets bounced off them, enters this and then goes here.
Different types of mirrors and lenses.
These are lenses. Huh?
Yeah.
So they focus the light.
Okay.
And in this black box here, we have a tiny larval zebrafish under the lens.
Here, this place, right?
Yeah. Under this lens, we have the tiny zebrafish, and that gets the light goes into the brain of the zebrafish, and then whatever the emitted light is there, that will then move into the detection path, and we have a camera here, which will take the images.
And then it sends to the computer.
Exactly.
So you are giving some stimulus to the fish here as well? Or?
Not right now, but that's the eventual idea. The same thing that they will do, that the electrophysiology.
So this is like something in process.
Exactly. It's still a work in progress.
Wow.
But we will do similar experiments to what they're doing.
Okay.
Yes. So if you want to look at some images and some single neurons, we can take a look. So this is the zebrafish brain. So there are some single neurons here. Can you see them? So the little
Like
rings.
these rings.
The rings are the single neurons,
Wow.
and we can go see it's moving a little bit.
How much have we scaled out? Like how much are we?
So
doing
this length is around 1 mm.
Wow.
So it is a big magnification. So the brain of the fish is just around a little less than 1 mm actually.
Okay.
So that's
there
how
in the
zoomed in.
previous
Yeah.
chamber they were just connected to one of these
Exactly.
neurons.
Yeah. So they were just looking at one of these tiny
And
cells.
the things that they connect to just one neuron.
Right. Yeah. So their needles are the
That
pipettes are so thin that they can go sit on one of these
Wow.
little rings and then look at what's going on inside it.
Okay.
So we can actually go through the parts of the brain
Sure.
here. So we have, if I move it up a little bit, so we have the forebrain here. This is the optic tectum, and
What's
this is
the optic tectum
it
like?
gets
Eyes?
visual inputs.
Okay, visual.
So the eyes in this fish would be here. Yeah,
Okay.
there's one eye here, one eye here.
So this part is related to the fish's vision.
Exactly.
Okay.
Yeah. And then this is the cerebellum. This
Okay.
region. That's what Shivangi probably talked to you about.
Like we have, right?
Like we also have. We also have a lot of these parts.
Okay.
And then we have the hindbrain here and the tail of the fish below.
Okay.
So that's what we
that's the front. That's the part that helps the fish see things.
Right.
And this is like the cerebellum.
And then the
this
tail,
is the
hindbrain and the tail of the fish. Yeah.
So a lot of the fish's body has the brain, right? A lot of the
No, that's okay.
yeah. So I thought I dropped something in the lab. So most of the body is the brain, is it? Like
What do you mean?
like most of the proportion in the body is mostly the brain, is it?
So this fish is around. This is 6 days old.
Okay.
So it will be around 3 or 4 millimeters long.
Okay.
And if you think about it, 1 mm of that is taken up by the
brain, yeah.
brain. Yeah.
Oh, okay. Nice.
So they are really small and yeah.
So right now we are seeing the fish that's like the.
Yes, that's under the.
It's actually a larvae, right?
It is a larva. It's alive. And
Okay, it's alive.
It's alive.
And where is it? It's swimming in some medium, right?
So I think Poonam showed you the restraint preparation where you fix the fish in agarose so it cannot move.
Yeah.
So the reason for that is because we have this
It's taken
microscope which is stationary, right?
Yeah, yeah.
So we need the fish to be in this one spot.
Yeah.
So we need to.
But I mean, how does it like take the nutrients and all? It's the medium that provides for that, right?
Yeah. So it's a very.
To make it like to stay alive, right?
So the gel that we use is mostly water.
Okay.
Which means that water, and it's also covered. The dish is covered with water,
Okay.
which means that the water can pass through the gel and go to the fish, and it can exchange oxygen, carbon dioxide, that sort of thing.
Okay. Nice.
So that's it has no problem staying alive. It can stay up to it can stay for more than 12 hours in the agarose.
Wow.
So you can do long experiments if you want.
Nice. Thank you so much.
Thank you so
Harsha.
much.
And with this, we end our live session. And today we learned that scientists, even from biological fields, have to know so much of physics and chemistry and they're even building microscopes on their own, like this really fancy one here. So yeah, so it's just like the theme we have today. Let's support it. Let's acknowledge it. Let's respect the interdisciplinary research happening not just in this institute but everywhere around India and across the world. So happy National Science Day to all of you from everyone here at NCBS. Hope you enjoyed our live sessions. If you would like to see more of them, do write in the comments. Bye.